everyone.
Hello.
Yeah, if you would type your name into chat for me, I'd appreciate it. Everybody had a nice weekend. Yeah, so if you wouldn't mind, just put your first name or your nickname into chat for me, so I have a record of who came today. Thank you. All right. All right. So we've had a little bit of an adjustment in our schedule. We're going to be doing protein catabolism 2 today, so we're about a day behind now. I have updated the schedule on Canvas, so if you ever need to pull it up, you can go to the home page on Canvas and open up the schedule from there. My dogs are going crazy because I have a bird feeder. I have a couple of them attached to a window, in my living room. Acrylic, lightweight bird feeders. And the neighborhood squirrel has figured out how to take a flying leap off of the back deck and land on the bird feeder. So he keeps doing it, and my dogs are going crazy. My dogs sit there and watch the birds come to the bird feeder. They love it when the birds come. But when the squirrel gets on the bird feeder, they get very upset. So hopefully you won't hear any barking, but if you do, you'll know why.
We have a squirrel relocation program here at our house.
That's what I need. I'm going to have
friendly
to
trap them and walk them pretty far away from our yard in a very nice spot.
This squirrel has been in the neighborhood for a little while, and he's enormous. He's as fat a squirrel as I've ever seen. And he's a bird feeder, genius. He figures out how to get in the bird feeders. He does it all over the neighborhood. And yeah, so he has discovered our bird feeder now. And like I said, he launches himself into the air to land on the bird feeder. And my dogs are having none of it. None of it.
I was going to say, if the dogs start barking, you should point the camera in that direction because it must be hysterical.
Oh my gosh. It's just funny to me that they don't bother the birds. When the birds come, the dogs are just sitting there quietly watching the birds, But when that squirrel comes, now that's another story. All right. All right. So before we get started on the lab exercise for today, I did want to just take a peek at our schedule. I also wanted to talk just briefly about a story that's been in the news that you may or may not have heard this weekend, but it actually relates to microbiology. So I thought I would mention it. Have you been hearing anything about the environmental catastrophe going on in Florida this weekend? Have you heard anything about this? There is a sort of a slow environmental disaster going on down there right now. Yeah, Vicky says yes. It's in a place called Manatee County, which is close to Tampa Bay. And what happened this weekend is of an old sort of defunct factory called Piney Point, which created several large lagoons full of toxic wastewater. One of those lagoons sprung a leak, and started to leak this toxic water, which is making a stream all the way to Tampa Bay. There was some concern over the weekend that the entire lagoon was going to fail. I'll show you an image of this thing in just a moment. And so there was terrible fear that if one of the walls of the lagoon failed, some 20 million gallons of water was going to head straight for a residential area, flooding out homes and possibly killing people. So every effort was made this weekend to encourage drainage out of the lagoon to prevent that catastrophic failure. But the problem is the water that is being slowly drained out of this lagoon is toxic. And here's where it relates to microbiology. The factory that used to be on this property, they used to be, one of the products that they made was fertilizer. I'm not sure if they're still making anything else, but at least in the past, they have been a manufacturer of fertilizers. And if you're not familiar, back in the old days, before the EPA was established, it was very common for factories and manufacturers, basically, when they generated wastewater in their manufacturing process, it was very common for them to simply let it out into the environment. Factories would install these effluent pipes that would just leave the factory, and they would just dump their wastewater into the surrounding environment. The EPA established laws that state that you're not allowed to release your toxic, poisonous wastewaters into the environment. But instead, what you can opt to do is dig this massive sort of a lagoon, a holding pond, if you will, and you can pump your toxic wastewater into that. You have to line these lagoons with some kind of a impermeable liner like plastic or rubber, but you are allowed to pump wastewater out into these ponds, basically these man-made ponds. The water then evaporates out of the pond, and the toxic, sludge is left behind. I was always curious what everybody thought was going to happen to the toxic sludge, but this has been legal for a long time now. Unfortunately, a lot of times in these holding ponds, the liners, as they age, they crack and they rupture. And this is one of dozens of cases of holding ponds that have sprung a leak because the liner was damaged over time. So it was noticed that water was leaking out of this holding pond. And again, the big concern was that the whole wall, the whole levee wall, was going to collapse. So over the weekend, efforts were made to sort of encourage the slow leak and discourage the whole thing from falling apart. I'll show you a picture of this place if you haven't seen it. All right, so let me show you this first because it might make a little more sense. This is what this place looks like from the air. So you can see there's just this massive holding pond. You can see the buildings right here. So there's 1, 2, 3 of these places where they can just dump their wastewater, basically. And it sits in the sun and the water slowly evaporates. Now, all of these are lined. These holding ponds are lined with an impermeable material, again, like plastic or rubber. And what happened was a leak sprung out of the side of one of these. So it's hard to tell, but this right here, this strip that you see that's very light colored, that's a roadway. So that's how big these are. And the walls that surround these holding ponds, they're like levees. They're tall. They're like levees, but they're earthen. So somebody noticed that this sort of tunnel developed. in one of these walls, and out was coming this effluent, this toxic water. Now, the reason this is interesting to us in microbiology is because of what's in this water. So the waste of fertilizer, the wastewater from a fertilizer plant, contains very high levels of phosphate and nitrate. And those are two molecules that will encourage the very rapid growth of microbes. Microbes can use both of those molecules as nutrients. As we've talked about, they can use phosphate and they can use nitrate in metabolic pathways, depending on the microbe. So as this wastewater has leaked out and started making its way downstream towards Tampa Bay, of course, all the people that make their living from the waters of Tampa Bay are very, very concerned, because these 20, 30 million gallons of water that is polluted with phosphate and nitrate are going to go into that bay and are going to set up for essentially a bloom of microbial life. Bacteria and archaea and algae and fungi and all those types of things will most likely grow very, very quickly as all of these nutrients become available, and the balance of the ecosystem in Tampa Bay will be disrupted. We've seen this happen before. This is not the first time in history that this has happened, that water, wastewater that is rich in phosphate and nitrate has entered a natural body of water. Unfortunately, we have historical examples of it. You get these massive blooms of microorganisms in the water body. Eventually, all those nutrients get consumed. The bloom dies off. Now you have gazillions of microbial cells dying all at the same time. And the other organisms that live in the water will be adversely affected by that. So fishermen, shell fishermen, people who run charter tours in Tampa Bay, everyone is very upset, and rightly so. Rightly so, because this is like I said, it's a slow motion catastrophe for that bay. So it's very, very sad, and it's frustrating because, again, it's not the first time that one of these holding ponds has given way. And we keep allowing these factories to use holding ponds for their toxic waste. And we need a different system. We need a system that's better than this. The governor of Florida was very, very happy to announce that the risk of a catastrophic flooding event for the people who live in that area, that is apparently past us now, because that large holding pond is already almost empty now. But he didn't talk at all about the environmental disaster of letting all that water out. Microbes are everywhere. Microbes live everywhere, and they live in natural bodies of water, and they're normally held in balance, like all living things are held in balance by things like how many nutrients are available, and how much room there is to grow, and what kind of predators are around for them, and so on and so on. And now basically they're being handed this soup of phosphate and nitrate. And people are afraid. People are afraid what's going to happen. Oh, he's back. Girls, that's enough. So I thought I would bring it up because it's very interesting how it relates to microbiology. If you're not familiar with the dangers of phosphate and nitrate in terms of, again, triggering one of these blooms of microbes, if you're not familiar with that, and many people in the public are not, you wouldn't realize how dangerous this kind of a situation is. So, I feel terrible again for the other creatures who live in Tampa Bay right now, because it doesn't look good for them over the next couple weeks. So I'm going to pull up our schedule. If I can find the right one. Let's see, this is our written schedule. This is again at the end of our syllabus. But as you can see, I've adjusted it now. Here we are right here on April 5th. Of course, you have a lecture exam today. I noticed that several of you have already taken it and already handed it in. Thank you. If you haven't yet gotten yours in, be sure to get that to me by this evening. We'll be doing the second part of protein catabolism today. And then looking ahead, you've got the immune system on your lecture schedule for the next couple of days. We'll be talking about respiration on Wednesday. That's actually our last day discussing these biochemical tests that we run in the laboratory. Next week, when we get together, on Monday, we're going to talk about the unknown microbe assignment that you're going to have. I'm going to talk about we'll take a look at the instructions for the assignment next week. We'll talk about how to use these tools called dichotomous keys. And I'm going to actually hand out the data to everybody so you can get started working on that. We are rapidly coming towards the end of the semester. So I'm happy that so many of you are doing well. Keep up the good work. We're almost there. So you're doing a great job. When I pull up our Blackboard page, oops, nope, try again. Let me try that one more time. Yeah, here's our Blackboard page. This is for this week. And then what you'll notice next week, I don't have it up here yet, but what I'll put up here for next week is all of the materials that you will need for the identification of the unknown microbe assignment. I'll put everything all together in a folder for you up on Canvas so you can find everything. But again, we'll take some time and talk about that together next week before you actually need to begin the assignment. All right. All right. Any questions about anything? Any questions at all? The other thing I found really interesting about the story out of Florida is apparently this factory has manufactured a few things over the years. And I don't know exactly why, but one of the waste ponds, I don't know if it's all of them or some of them, but apparently radium is in at least some of those ponds. So if you don't know what radium is, it's a radioactive material. The thought that is sitting in one of those ponds is horrifying. But there was some concern early on that radium was actually being released into the environment. Apparently, when the governor was asked, what about the radium at a press conference, his answer I found very unusual. He said, and I'm quoting now, he said, the water that is coming out of that pond is not radioactive. And he said it very deliberately like that. And I remember thinking to myself, well, that's exactly right. Water is not radioactive, but radium is. So the real question is there radium in the water that's leaking out of that pond? And he wouldn't answer that question. So I'm going to hope that there was no radium in the pond that leaked. That would be 100 times worse. But only time will tell. Only time will tell. All right. So when we were together last time, we started talking about protein catabolism. And if you remember, we were talking about how microbes often break down protein to get at the amino acids in the protein. And we said sometimes it's also capable, or it's also possible for some microbes to take amino acids and break them down in order to get at the carbon that's present in the amino acids. So what we're going to be doing today is looking at a few additional tests around protein catabolism, including tests that will look for this kind of metabolic activity for microbes that are breaking down amino acids, catabolizing amino acids, instead of using them to build more protein. So our objectives for today, we're going to define a couple of new pathways that we haven't talked about yet. One of them is called deamination and one's called decarboxylation. There's actually a third pathway called desulfuration that we'll also mention. That desulfuration pathway is going to result in the production of what's called hydrogen sulfide gas. So there are tests that we can use, as we've done with all the other metabolic pathways we've talked about. There are tests that we can use in the lab to see if a microbe is performing any of these pathways. Specifically, we're going to look at two tests of amino acid catabolism. One is called phenylalanine deamination. Phenylalanine is one of the 20 amino acids. And cysteine desulfuration. Again, cysteine is the amino acid here. So remember, most of the time that microbes and other cells catabolize a protein, break apart a protein, they're going to break it apart to get to the amino acids. They're going to set those amino acids free, and they're going to use them for anabolic processes to build brand new proteins. And you and I do the same thing. We consume protein in our diet, and we have certain digestive enzymes that break down the protein into all the amino acids that are present in the protein. And then our cells can take those amino acids and use them to make new proteins. Very, very common pathway. But it's also possible for cells to take an amino acid and break that apart in order to get to the carbon skeleton in the amino acid. So let's look at these processes for breaking apart amino acids. The first pathway is called deamination. We'll take a look at amino acid structure again in just a minute, just to remind ourselves of what amino acids are built of. But part of every amino acid is what's called the amino group. So deamination is simply the removal of the amino group from an amino acid. When the amino group is removed, the product that's going to be produced is an acid and ammonia. It's actually two products that will be left over when an amino acid is deaminated. There's a whole big family of enzymes that perform this deamination pathway. Now, there's another pathway called decarboxylation, where the carboxyl group from the amino acid gets removed. And the product molecules are going to include a base and carbon dioxide gas. That base gets a name. Depending on the amino acid, it's going to be an amine base. And again, a whole family of enzymes are going to do decarboxylation. Now, desulfuration is different because in desulfuration, there isn't a group, a sulfur group on an amino acid. As I said, we'll take a look at a model of an amino acid in just a moment, but we know there's an amino group on every amino acid. We know there's a carboxyl group on every amino acid. We know there's a single hydrogen atom on every amino acid, and we know there's something called the R group. And the reason one amino acid is different from another is because the R groups are different. The functional groups are different from one amino acid to another. Sometimes, not always, but sometimes amino acids will contain sulfur in their R group. For example, the amino acid methionine and the amino acid cysteine both have sulfur as part of their R group. So there is no sulfur group, but some amino acids do have an R group that contains sulfur. And bacteria that are capable of it can do a reaction called desulfuration. They remove the sulfur using an enzyme called desulfhydrase. And that sulfur will then be combined with hydrogen to create hydrogen sulfide. So some microbes can perform all three of these pathways. Some can perform one or two. Some can't perform any of them. It depends on the species. Another thing to note, when we talked about microbes breaking down protein to get to the amino acids, we talked about the microbes secreting those enzymes, right? For example, when we talked about the gelatin hydrolysis test, we said that certain microbes can secrete these enzymes called gelatinases out into the environment. And if there's gelatin out there, the gelatinases will break it apart into the individual amino acids. Those kind of enzymes are exoenzymes. The microbe is secreting the enzyme out into the outside world, out into the environment. Those enzymes break down the protein, and then the microbial cell takes up the amino acids, absorbs the amino acids. Now, when it comes to breaking down amino acids themselves, that's a very different situation. If a microbe takes in amino acids from the environment and then decides to break apart those amino acids, the enzymes that do that work, those are all endoenzymes. Those are all inside the cell. So breaking protein down into amino acids, that's done outside the cell primarily with exoenzymes. Breaking down the amino acids themselves, that's happening inside the cell with endoenzymes. On this slide, you can see a drawing of just a generic amino acid, because all amino acids have the same basic structure. Remember, there are 20 of them that are common in nature. Microbes build protein using those 20 amino acids. You and I make our proteins with those 20 amino acids. It's the same types of amino acids that you see used to build proteins all across all the living cells. So about 20 of them, some of them human beings can manufacture, and some of them human beings have to eat, have to consume. There are 8 amino acids that are what we call essential in the human diet. You have to eat them in your diet because your cells can't manufacture them from scratch. But the other 12, your cells can manufacture from scratch. So basic amino acid structure has a central carbon atom. And like all carbon atoms, it can form 4 single covalent bonds. So one of those bonds is going to be to a single hydrogen atom. One is going to be to an amino group. One is going to be to a carboxyl group. And one is going to be to this R group. And remember, all that R stands for is some kind of functional group. So depending on what the R group is on an amino acid, that's what makes it different from all the other amino acids. And there are lots of R groups that you find in amino acids. I've got some of them here on this slide. Here's a carbonyl group. Here's a carboxyl group. Here's a phosphate. Here's a hydroxyl. Here's a sulfhydryl. There's all kinds of functional groups, R groups, that are attached to amino acids and give them their individual chemistry. Remember, it's the R group of an amino acid that's going to determine how it's going to behave chemically. So in a long string of amino acids in a protein, the order of the amino acids is going to determine how that protein folds up. If two amino acids are sitting side by side and they both have a polar R group, those are going to be attracted to each other. So those two amino acids will get closer together. If one of them has a polar R group and another one has a non-polar R group, they're going to be repelled from each other. And they're going to move farther apart. There are acidic R groups and basic R groups. So depending on the chemistry of the R group and the order of the amino acids, that's what dictates how a protein folds up, where the hydrogen bonds are going to be, and where the van der Waals interactions are going to be, and so on and so on. All right, any questions so far? Any questions about anything I've said so far as I'm on squirrel patrol? All right, if there are no questions, what I'll do now is we'll move forward and look at these particular tests for amino acid catabolism. So the first test is called phenylalanine deamination. So remember, the deamination just means that the amino group is going to be removed and the product is going to be an acid and some ammonia. All right. The particular amino acid that we're going to use in this test is the amino acid called phenylalanine. Now, the phenylalanine deamination test uses an agar that is just a basal culture medium. It's nutrient agar. The only thing that's unusual about it is that it's got a lot of phenylalanine in it. So we're going to add phenylalanine into the nutrient agar. We typically do this test in a slant. Now, it's going to show whether or not this microbe can use this deamination pathway to catabolize phenylalanine. Down here, you can see I've listed the name of the acid. It's actually called phenylpyruvic acid. Don't worry about that name. I'm not going to ask you that name on an exam. Just know that an acid gets produced and ammonia gets produced. Now, this is one of those tests where after you inoculate the slant tube and you put it in the incubator to incubate, when you take the tube out of the incubator, you have to add a reagent to it in order to see whether you've got a positive or a negative result. In this case, the reagent is called Ferric Chloride. And if you're not familiar, that word ferric refers to iron. So this is a reagent that contains iron. Now that iron is going to react with phenylpyruvic acid. It's going to react with the acid in the tube if it's present. And it's going to result in a color change. It's going to turn the media a dark green color. So that's your positive result. If the microbe has the ability to break down phenylalanine using the deamination pathway, it's going to produce this acid and some ammonia. When we take the tube out of the incubator, when we add in the ferric chloride, it's a liquid that you just drip in to the slant tube. When we add the ferric chloride, if there's any phenylpyruvic acid in that tube, It's going to react with the iron, and you're going to get a dark green color. If you drip the ferric chloride into the tube and nothing happens, you don't get a color change, that's a negative result. That means that the organism was not able to break down the phenylalanine. On this slide, you can see a couple of these slant tubes. You can also see that the ferric chloride has been added to the tube. So this is the color change you're looking for right here, this dark green color. Now, this is a slant. Remember, this is solid media in here. It's agar. So when you go to inoculate this, you're just going to make a streak down that slanted surface of the agar using your sterile loop. You're going to put it in the incubator and allow it to incubate for 24 to 48 hours. When you take it out of the incubator, the tube is going to be this color. It's going to be this beige color, very boring, nutrient agar beige. Then you're going to add the ferric chloride. It's this liquid right here. It's sort of a yellowy color. It looks kind of brown here, but it's usually a yellowish color. When that hits the cells, it's going to indicate anywhere that they have released the enzymes to break down the phenylalanine, and you're going to get this green color. So it's not unusual for the green to just be up in the top part of the tube. These cells, remember, they're growing right on the surface of the slant. And when they release the enzymes when they absorb the phenylalanine from the agar, and they produce that acid and the ammonia, it's the acid that's going to react with the iron in the ferric chloride and cause that green color change. Over here, this is what a negative result would look like. It's harder to see the slanted surface on this tube, but it's right around here. And this brown yellow liquid right here, that's the ferric chloride that got added in. And you can see there's no color change. So this is a negative result. Now, the other test we're going to look at today is this desulfuration test. You can use any amino acid that contains sulfur in its R group. And we're going to use cysteine. People typically either put cysteine in here or methionine. And we're going to use cysteine in ours. So if the organism has the enzyme necessary to break down cysteine, to desulfurate the cysteine, what will happen is the cysteine that gets released will result in the production of hydrogen sulfide. Now, this is an interesting test because it uses a medium that we haven't talked about yet, but it's actually a medium that gets used for several different tests in the laboratory. Now, we haven't talked about this medium, so I want to say a couple of things about it, but it's referred to as SIM media. You can use SIM media as a broth, you can use it as a slant, you can use it as a deep, whatever your needs are. We're going to look at it in a slant. When the cysteine gets desulfurated, when it gets broken down, what's going to be left over is an acid, pyruvic acid, as it turns out, and hydrogen sulfide gas. So remember, we said for phenylalanine, what's left over is acid and ammonia. For this one, it's acid and hydrogen sulfide. And in this case, it's not the acid that's going to react with the reagent, but the hydrogen sulfide that's going to react with the reagent. We're going to put in another iron-containing reagent here, another ferrous, but this time it's ferrous sulfate. For this one, it was ferric chloride, and for this one, it's ferrous sulfate. It's the iron that matters in these. When hydrogen sulfide combines with this iron-containing reagent, you're going to get a black color change. You're going to get a black precipitate. So if the microbe used the cysteine for desulfuration and released the sulfur from the R group with those enzymes, what's going to happen is hydrogen sulfide will be produced in the agar. And when that hydrogen sulfide meets that reagent, the ferrous reagent, you're going to get a black color produced. So I said I just wanted to say a few words about sim media because it is a media you should be familiar with, even though we didn't look at all of the tests that it's used for. This word sim stands for sulfur, indole, motility. So this is a sort of an all-purpose media because you can use it for several different tests. We're using it for this sulfur reduction reaction. We're using it to test for desulfuration. But you can also use it to look for the production of a metabolite called indole. And you can also use it to look for motility, just general motility of an organism. So take a look at the recipe for sim media. Normally it contains peptone. It contains a pancreatic digest of casein, and all that is a partially digested form of milk protein. So it has two sources of protein in it, partially digested milk protein and peptone. It also contains an iron compound called ferrous ammonium sulfate, and it contains a sulfur source normally called sodium thiosulfate, as well as agar. Because again, if you want to use it as a solid medium, you're going to have to add agar. If you want to use it as a broth, you don't need the agar. So we're going to use this medium, but think about what we're trying to do here. We're going to add cysteine into the media because we want to see if the microbe can break down the cysteine. So we want any sulfur to come from the cysteine. So we're going to have to take this out of the medium. We're going to remove the sulfur source because we're going to add in a different sulfur source. We're going to add in cysteine. The other thing you have to remove from the sim media is this. You have to remove the iron containing material from it because we don't want to add the iron in until the next day. So when you're using sim media to do cysteine desulfuration, you have to take the iron out, you have to take the sulfur out, and you have to add in your own cysteine. So let's take a look at what these tests look like. Over here on the left-hand side, these are two cysteine desulfuration broth tubes. And over here, you can see solid agar, cysteine desulfuration agar. So there's nothing wrong with using the broth form. In fact, some people like to use broth because when you add the ferrous sulfate in after incubation, you're going to get the black color change all the way through the broth. So it's very easy to see. In other words, you're going to take this broth tube, you're going to inoculate it with your organism, you're going to put it in the incubator, let it incubate, then you're going to take it out the next day. You're going to add in this reagent called ferrous sulfate. That's this liquid you can see sitting on the top of this tube. And then you're going to look for a color change. And again, if you're using a broth, that black color will show up all the way through the broth. So that color is telling us that there's hydrogen sulfide in this tube. The iron has reacted with the hydrogen sulfide, and the hydrogen sulfide was produced when the microbe broke down the cysteine using desulfuration. Now over here on the right side, these are the agar tubes. You can see they're agar deeps. They're not slants. You can use a slant or you can use a deep. And remember, usually when we're using a deep, it's because we're trying to grow an organism that doesn't really like oxygen, right? That doesn't grow well in the presence of oxygen. Remember, when we inoculate a deep, we use a wire and we stab the media. So this is what the tube would look like before you inoculated it. And then after you stab it and put it in the incubator, when you take it out and you add the ferrous sulfate, sometimes you will see this happen. That's a positive reaction. There is black color all the way down that stab line. Remember, you stab the agar, you're putting cells deep down into the agar from that wire. And those cells are going to grow on that stab line. And when you add the ferrous sulfate the next day, if there's hydrogen sulfide in there, it's going to turn black. So it's a little harder to read when you use it as a deep. You have to be able to look into that stab line to see the black color, but it's there. All right, so 2 tests. Phenylalanine deamination, and we're looking for a green color change. And cysteine desulfuration, we're looking for a black color change. Any questions about this so far? There are other tests that you could do. You could look at something other than phenylalanine for deamination. You could look at something other than cysteine for desulfuration. It has to have sulfur in it. It has to be one of the amino acids that has sulfur in it, but you could choose any of them. You could also do a decarboxylation test. We're not looking at that test today, but they exist as well. And again, what you're going to learn is some traits about your organism. What traits does it have? What's it able to do metabolically? Because that helps us identify different types of microbes. All right. So our organisms for today include E. coli, Enterobacter aerogenes, and Proteus vulgaris. And we've seen these organisms before. Our good friend E. coli, gram-negative rod, fecal coliform. Enterobacter aerogenes, another gram-negative fecal coliform. And then Proteus vulgaris, which is an opportunistic pathogen. It's a gram-negative rod like the other two. It's also a fecal organism. It's an intestinal or an enteric organism, but it's not a coliform. It does not ferment lactose. The pathogenic strains, in other words, the Proteus vulgaris that acts as an opportunistic pathogen, can cause all kinds of problems. It's very common in a common cause of UTIs, urinary tract infections, certain respiratory infections, sepsis, and again, burn infections. It's a real problem in burn patients. And here are our results. These are the phenylalanine deamination tubes. You can see that the agar is in a slant formation in the tube. These tubes were inoculated with E. coli, Proteus vulgaris, and Enterobacter aerogenes. Here's the ferric chloride, excuse me, that was added in after incubation. This little yellowy gold liquid. You can see that for E. coli, nothing happened when you added the ferric chloride inside. No color change. No color change for Enterobacter aerogenes either. If you remember from a previous lab, E. coli and Enterobacter aerogenes are kind of hard to tell apart. We talked about some tests in a previous lab that we can use to help us distinguish these two. They're both coliforms. They're both intestinal. They're both gram-negative rods. they have a lot of characteristics in common with each other, so they can be a little hard to tell apart. In this case, phenylalanine deamination isn't going to help us because neither one of them is capable of it. But Proteus vulgaris is capable of it. Proteus vulgaris has the enzymes necessary for that deamination reaction. And you can see this green color change. Notice how it's only right along the surface of the slant. It's very interesting. It hasn't really gone down into the agar at all. It's only going to react with the acid that has been produced so far. So this is another one of those times when if you put this tube back in the incubator and allowed it to metabolize longer, allowed it to further break down the phenylalanine, this green color would go deeper and deeper and deeper down into the tube. All right. Here is our cysteine desulfuration reaction. This is the broth again. All 3 broth tubes. This is SIM broth that has been, again, modified. We took the sulfur out, we took the iron out, and we added in a bunch of cysteine because we want to see if the microbe can break down the cysteine. All 3 tubes were inoculated. Here's the E. coli tube. Here's the Proteus mirabilis tube. Here's the Enterobacter aerogenes tube. Now, if you look, you can see there's like some liquid on the top. Then there's this sort of a white line here. And in the E. coli tube, you can see there's a white line going down into the broth too. This is one of those strings that we've talked about before, strings of cells growing down. Over here, the string is really thick, sort of a Y shape. Again, there's some cells growing on the surface of the broth. That's the white line. Here's that liquid that we added in, the ferric sulfate. You can't really tell what's happening in the Proteus tube because it's turned black, but you can see some cells growing on top. That's the white line. And again, the ferric chloride liquid. Now, I want to make an important point here. This is cysteine desulfuration, this test, using SIM media as a broth. This is not thioglycolate medium. So even though you're seeing some growth patterns in there, you're seeing strings hanging down and you're seeing cells growing on the top and things like that, you can't evaluate that pattern with this media. Remember, if you want to evaluate what oxygen category that an organism belongs to, you're going to want to use thioglycolate medium. Remember, that one is kind of semi-solid. It's a whole different medium. This one is not designed to evaluate the pattern of growth. You can determine whether or not something grew in the tube. That's good to know, right? It's good to know that E. coli can grow in that tube and Enterobacter can grow in that tube. That's good. But the change we're looking for in this test is the color change. And if the tube turns black when we add the ferrous sulfate. So only the Proteus test is positive here, okay? All right. And again, because it's a broth, the positive result is very obvious. So the reason this tube turned black is because we added in an iron-containing reagent and the iron reacted with the hydrogen sulfide in here. No hydrogen sulfide, no black color. So only Proteus mirabilis was able to take the cysteine and catabolize it, break it apart, and produce an acid and hydrogen sulfide. And we can see the hydrogen sulfide by adding iron to the tube. These two, they grew in the tube, but they're not able to break down the cysteine. It's just not one of their characteristics. So again, our test results show that Proteus Vulgaris, there are a couple of interesting features of Proteus Vulgaris, right? It can break apart, it can deaminate phenylalanine, and it can desulfurate cysteine. Very, very interesting organism. We would not be able to use these results to tell E. coli and Enterobacter apart because they are negative for all of it. But if we thought we might have a Proteus, these are two tests that would be very helpful to us. All right. So the next time we get together, we're going to be talking about your unknown microbe. Actually, I should double check that. I don't know if I said we were going to do that next time or next week. But we are going to talk about respiration as our last biochemical test. So we'll talk about respiration, and we do that with two tests. One is called the oxidase test, and one is called the nitrate reduction test. And then after we finish discussing those tests, you'll be ready. You'll be ready to look at a whole table of test results from all of these sorts of tests that we've been talking about, and use those test results to identify a microbe based on the results. And I'm going to give you some of the tools that you'll need. I'm going to give you the data, and I'm also going to give you some keys, some what are called dichotomous keys to help you go through that data and help you identify what microbe you've got. Excuse me. All right. So questions about what we talked about today? Questions about deamination or desulfuration? Any questions about what we're looking for or what the color means? These tests are fairly straightforward. You do have to add your reagent though when you take that tube out of the incubator. If you take the tube out and you forget to do that, you're going to think you have a negative when you may or may not. All right. So I'm going to, just for my own curiosity, because I've already forgotten, I'm going to pull up this. Yeah. So next time we're going to do the respiration material. And then next week, we will talk about the unknown assignment. So for Wednesday, you've got your lab slides if you need them. And then remember this week for homework, it's only going to be protein catabolism because you didn't do any you didn't do any homework on the first half of protein. So I saved it for this weekend. So for Sunday at midnight, there's lab questions only about protein catabolism this week. All right. Any questions at all? Anything I can help you with?
Actually, Dr. P, my husband just fell and gassed his eye open, so I have to run to the ER. I sent you a private chat message, but I just wanted to let you know I'm leaving right now.
I'm so sorry to hear it, Suzanne. Please go.
Everything happens to me.
Give him my best. I hope there's no one in the ER but you guys.
Well, we go to the VA, so that's a good bet.
Okay.
Thank you.
All right. We'll see you next time. Anything else? All right. If you haven't already, don't forget about your exam. Okay. Make sure to get that to me by tonight. And I will see you all on Wednesday. All right. Have a good evening, everybody.
Thank you.
You're welcome.